Okay, I have to admit, woo, that's loud. Um, every time I've seen everybody wear these microphones, I don't know for those of you who are as old as I am, but back in the like 80s workout videos, I feel like I should be like, or given a TED talk or a motivational speech here. Anyway, but we're going to go ahead and get started with the next session. And so I want to welcome you to the session on sarcoma treatment and motion, new therapies, trials, and approvals. And I would like to introduce our speakers. For those of you who don't know Robin, he is a medical oncologist specializing in the treatment of bone and soft tissue sarcomas and head of the sarcoma unit at the Royal Marsden in London. He is also the principal investigator of the Sarcoma Accelerator Consortium and the current president of CTOS. Dr. Jones trained in medical oncology at the Royal Marsden, and between 2010 and 2014, he was head of the sarcoma program at the University of Washington Fred Hutchinson Cancer Research Center in Seattle, Washington, in the United States. He has experience in conducting phase 1, 2, and 3 trials, as well as translational studies in sarcoma, as well as laboratory-based studies. I also found out at dinner last night, according to the Sarcoma Cancer Ireland team, he's a sarcoma god. But before I invite Robin to the podium, we have a video from Gerry Feeney, a patient who joined one of Robin's trials and talks about his experience finding treatment options after several recurrences. Gerry, unfortunately, is not with us in Stockholm, but shared his story via video.
Hello, everybody. My name is Gerry Feeney, and I'm a sarcoma patient, myxofibrosarcoma to be precise. I'm sorry I can't be with you today, but I'm still recovering from recent surgery, so the travel would be a bit of a challenge. What I wanted to share with you today is my experience under the care of Professor Jones and how he enabled me to access some innovative cancer treatments. My sarcoma story goes back to 2017, when an odd but painless lump appeared on my left leg. It was initially misdiagnosed as a lipoma, then as angiosarcoma, undifferentiated pleomorphic sarcoma, and finally as myxofibrosarcoma. Along the way, I've had 2 courses of radiotherapy and 6 operations before the recent surgery, which was my 7th. I was treated for all the operations at the Nuffield Orthopaedic Centre in Oxford, part of Oxford University Hospitals, but was referred to the Royal Marsden in London when I presented with my 7th recurrence early last year. The reason for the referral was for an isolated limb perfusion, a technique whereby the affected limb is isolated for chemotherapy treatment. However, I was considered unsuitable for the ILP due to the extensive prior surgery. So at that point, Professor Jones intervened and put me forward for an early stage trial of an oncolytic virus. Now, this was a bioengineered so-called cancer-busting virus, which was intended to attack the tumours in my leg while also creating a systemic immune response, similar to a vaccine. The phase 1 trial was intended to roll out in 2 parts, where part 1 was to have the synthetic virus administered as a monotherapy, and part 2 was to combine the virus with an immune checkpoint inhibitor, that is an immunotherapy drug. The objective for the combination was to alter the characteristics of the tumour, a process known as oncomodulation, in order to make the virus-altered tumour more responsive to immunotherapy. And this is important for sarcoma, which tend to be cold tumours, generally seen to be unresponsive to immunotherapy. And I was a patient in part 1 of the phase 1 trial, which involved 4 direct injections of the synthetic virus into one of the more accessible tumours in my leg, after which I was admitted for an overnight stay in hospital for observation. The injections are administered at fortnightly intervals. Unfortunately, my tumours did not respond to the virus treatment. Professor Jones then arranged for me to have an off-label immunotherapy treatment. This treatment comprised a combination of ipilimumab and nivolumab. These are CTLA-4 and PD-1 immune checkpoint inhibitors, respectively. Ipilimumab and nivolumab, known as Ipi-Nivo in the trade, are proven treatments for cancer such as melanoma, as well as others. Ipi/Nivo is not licensed by the UK or EU regulators for sarcoma, but is accessible under compassionate access if the patient has limited treatment options. Professor Jones arranged for me to have Ipi/Nivo provided by Bristol Myers Squibb under their pre-approval access scheme. Other patients with myxofibrosarcoma have responded well to Ipi/Nivo, hence the rationale for arranging off-label access to the treatment. However, immunotherapy, like chemotherapy, can cause unpleasant side effects, and in my case I experienced a grade 2 immune-related adverse event, which unfortunately meant that the treatment had to be halted. Professor Jones and his team tried to identify another trial which might be appropriate for my indication, but no suitable trials were recruiting patients at the time, so I was referred back to Oxford for surgery. I would nevertheless like to thank Professor Jones and his team for their efforts arranging access to the trial that I did have, as well as the off-label immunotherapy. And although further surgery was subsequently needed, the systemic treatments at the Royal Marsden may well be instrumental in preventing further recurrence and metastasis. So with that, I'd like to thank you for your attention, and I hope to see you all at a future SPAGN event when I'm fit to travel again.
And now, Robin, the sarcoma god.
No.
I'm just teasing my colleagues here from last night.
So I think one of the things for me is to just say how inspirational and meaningful it is to be here. Of all the meetings that I attend, I think that this really is the most inspirational, to be with all of you and to be able to sort of communicate and to talk to you about treatments, the challenges, but also the successes. And I've already warned Marcus that I'm not going to show many slides, but that I'm going to basically throw it out to you guys for, to ask questions and to discuss some of the issues. And I'd also like to thank Gerry for his presentation. And I think that really does show the challenges of treating some of these really rare sarcomas where we don't really have good treatments. And, we have to be inventive, imaginative in terms of the ways that we use the treatments that are available to us. And, in certain countries, and Sandra knows this well, in the UK, we're very limited in terms of being able to use off-label treatments. So, as I say, the fundamental message from this is that it's really inspirational to be here, and I'm really grateful for the invitation. So this is just a sort of outline, and I'm going to start with a brief introduction. And I suppose in terms of new trials and approvals, it's really a case of the best of times and the worst of times. There are positive clinical trials. There are approvals in process for sarcomas, but also over the last few years, there have been a number of negative trials, particularly in retroperitoneal liposarcomas. And thank you for mentioning the Accelerator Consortium, which is a CR UK, Sarcoma UK, amongst other grant agencies, a consortium between our centre in the UK, other centres in the UK, and also Italy and Spain. And the major thing about that is that these negative clinical trials, as we discussed yesterday, in terms of not only publications, that we should have outputs and a record of a negative clinical trial so that we don't do the same thing again, but we also need to learn from those negative clinical trials in terms of why some patients benefit and others, unfortunately, don't. So, as we all know, the fundamental problem for us as oncologists and those trying to develop new treatments in sarcomas is the profound heterogeneity, that there are so many different types of sarcomas, and each one of these is completely different. And that really does present a huge challenge in terms of drug development, with over 80 different subtypes. And I know Judith was here earlier on. I think in the next WHO output, there are probably going to be over 100 different subtypes. So it's almost a microcosm of all solid tumors. And that really does present challenges when some of these diseases are so incredibly rare. And of course, for oncologists managing metastatic or advanced soft tissue sarcomas, there are a number of treatment options available. And I suppose this is one of the sort of messages that I want to make today, that even in an environment where we have limited access to treatments, it's important that we try and think out of the box and use all of the treatments available. So Gerry mentioned isolated limb perfusion, which can be a very useful technique of just delivering chemotherapy to a specific area of the body, so the arm or leg. If the disease is limited to, say, the lower leg, it can be a very effective treatment. But as Gerry mentioned, unfortunately, in some situations, due to previous treatment, it may not be feasible. And we've had some patients where that has resulted in durable benefit and durable response, particularly in aggressive diseases such as angiosarcoma and also pleomorphic sarcoma. But as I say, there are a number of treatments available, and it's really, really important that we think out of the box and try and use all of these treatments in a tailored way for the individual patient. So in terms of drug approvals, I think the sort of poster child in many ways is gastrointestinal stromal tumor, with a number of positive trials and a number of drugs being approved, including avapritinib and ripretinib. And I think the thing here is to emphasize the importance of clinical trial involvement, and to be proactive in terms of offering clinical trials to patients, even in earlier lines of therapy. And that was one of the reasons that we had this discussion with Gerry regarding using clinical trials as an earlier form of treatment, rather than just going for conventional doxorubicin, which we know in certain sarcoma subtypes has very limited activity. And just to show the sort of difference that this can make for an individual person, this was a gentleman referred, and I told everybody my eyesight is really bad, referred to us back in July 2016 with this huge abdominal tumor. You can see that it measures over 40 centimeters, and had received standard treatment with imatinib, sunitinib, and regorafenib. And he went on to the phase 1 trial, and basically, eventually, the tumor shrunk down to about 11 centimeters, and then went on to have surgery with Dirk Strauss, and he remains on the drug to this day, 10 years later. So I think, again, this is really, really important that patients have access to clinical trials, have access to off-label medications, so that we can try and tailor the treatment to the individual. And of course, there are a number of other ongoing trials in GIST which will lead to approvals. And I'm not going to make this some sort of exhaustive list of all of the available clinical trials, but just to say that this is a developing field, and it's likely that there'll be a change in the treatment paradigm and more individualized treatment for patients over the next few years, including the use of ctDNA. And one of the newer drugs, bezaclastinib, there's going to be a presentation at ASCO this year based on the randomized phase 3 trial, which was in the second-line treatment of GIST. Patients having had treatment with imatinib were randomized to receive either the standard treatment sunitinib or sunitinib plus bezaclastinib. And the press release from last year showed that there was a significant benefit for the combination, both in terms of the median progression-free survival, so the time from starting treatment to the cancer progressing, but also in terms of response rate, in terms of the shrinkage of the tumors. And one of the other notable positive trials is the first ever positive randomized trial in chondrosarcoma, which was presented at CTOS last November. So chondrosarcoma being one of these tumors that's essentially resistant to conventional chemotherapy. And this drug, azikibart, Inhibrx 109, showed activity in early stage clinical trials, which led to the randomized phase 2 trial. And basically, this drug is meant to work by inducing cell death in the cancer cells. And a fairly simple design in terms of people being allocated to either the drug or placebo, with crossover on progression if they were in the placebo arm. And the trial was positive, so it showed a significant difference between the two arms. And in many ways, this was a modest benefit, but that's in the context of a disease that is, as I say, essentially resistant to all the treatments that we have. And also that one of the requirements for trial entry was that there had to be disease progression prior to entering the trial. So all these patients had disease that was progressing prior to going into the trial. And that benefit was maintained. One of the major things to look out for with this drug is liver toxicity, and the trial was adapted due to liver toxicity in terms of entry criteria, very stringent liver function and past medical history entry criteria to exclude patients that could potentially develop liver toxicity. A number of patients with chondrosarcomas also have tumors that harbor IDH1 mutations, and there's an ongoing randomized trial of an IDH1 inhibitor in conventional chondrosarcoma as well. And again, just to highlight the importance of clinical trial participation, this is a young woman who basically had an inoperable chondrosarcoma that was referred with progressive disease and symptomatic and radiological progression, but entered a phase 1 trial back in 2020 and continues on the treatment with symptomatic improvement and slight reduction of the tumor. And then a mention about desmoid tumors. We had a very good discussion yesterday in the desmoid tumor session. As we discussed, there are a number of promising agents in development for this disease, which is absolutely fantastic for patients and the community. One of these is AL102, which is a gamma secretase inhibitor, and is there's a biological rationale for using this type of treatment in desmoid tumor. And of course, there's already one approval in this class of nirogacestat, which was approved a number of years ago. But again, this drug has shown activity from early stage in terms of dose selection, and it's notable that there's really durable responses, and that the responses are very early in terms of benefit for patients. And as you can see from this chart, the majority of people have tumor shrinkage with this drug. And in terms of side effects, the main side effects tend to be skin, gastrointestinal reactions, but in general, relatively well tolerated. And this randomized trial is also going to be presented at ASCO this summer. And just a word regarding NTRK tumors. So these rearrangements, these molecular abnormalities are relatively rare, but we've treated about 7 to 8 patients at our hospital, and it can have such a profound difference in terms of the impact on patients' lives. And again, there are really compelling supporting data for the use of these drugs in patients with NTRK rearranged tumors. And as you can see from this chart, the vast majority of patients have tumor shrinkage and very durable responses. I have a number of patients. There's one patient who's been on larotrectinib for 5 years with metastatic spindle cell sarcoma with very minimal toxicity, having had disease progression on all conventional treatments. So again, for those individual patients, this is absolutely critical in terms of selecting treatment and tailoring treatment for people. But of course, there have been a number of negative clinical trials over the last few years, and most notably, as I mentioned, MDM2 inhibitors in liposarcomas, but there are a number of others listed on this slide. And I think one of the things is that we should really learn from all of these trials and try and understand, as I say, why some of these trials didn't quite meet the primary aim, but there's clear evidence of benefit in a subgroup of patients. And the unfortunate thing right now is for these trials listed here, we don't know why some patients benefited. And the role of MDM2 inhibitors in retroperitoneal liposarcomas in particular is really challenging, and the challenges of actually documenting radiological response. So the tumors are big, they're heterogeneous, and resist. So the standard way of just trying to measure one dimension doesn't really work that well in this particular disease. And in our consortium, working with colleagues from the UK, Spain and Italy, we're collecting radiological, clinical, pathological, as well as molecular data on a number of sarcoma subtypes, including retroperitoneal liposarcomas. And one of the things that we really want to do with this information, so thousands of patients, is to look at the radiological response in retroperitoneal liposarcoma to try and develop better ways of assessing response and improving from this sort of unidimensional RESIST methodology. And this just gives an outline of the work packages within the program, of course, working with my friend and colleague Paul Huang. And just really finally, I think I've gone on far longer than I wanted to, but in terms of other approaches, and that's where Gerry's presentation is so useful, I think that it is really important that we try and think out of the box. And there are a number of very rare sarcomas where we don't really have a standard treatment. And one of those is clear cell sarcoma. And again, this is so incredibly rare that we don't really have any prospective data regarding the response to doxorubicin-based chemotherapy. And this highlights the importance of collaboration, working together to actually document that anthracycline-based treatment doesn't work that well in this disease. And my argument is always that we should consider clinical trials and other approaches for patients with these types of sarcoma that we know that doxorubicin and other conventional chemotherapies don't work that well. But, again, coming back to this point regarding being imaginative and working outside of the box, it's important that we use other modalities. So we have a number of patients with pleomorphic sarcoma where we've used isolated limb perfusion with checkpoint inhibitors. In terms of phase 1 clinical trials, we're very fortunate to have a really busy phase 1 unit at the Royal Marsden where we can offer treatments to patients, particularly if there is a molecular rationale or if there is no standardized treatment. And of course, Gerry mentioned the compassionate access medication as well, which again, for a number of patients is really, really important. And, as I say, the environment in the UK is challenging, but there are ways of working around it. And one of the anecdotes that I've got is of a ctDNA study that we've got open at the Royal Marsden. There was a patient who'd been on avapritinib for metastatic GIST for about 8 years, but developed new liver metastases, and we biopsied these, and it was metastatic carcinoma. And he entered the ctDNA study, and there was another molecular abnormality, so an ALK rearrangement. And we've started him on an ALK inhibitor, and he's had a tremendous clinical and radiological response to that treatment. So again, trying to think out of the box of ways that we can use the available treatments to help patients. And this is a patient who was on a phase 1 trial of a checkpoint inhibitor plus gemcitabine. And this is a patient who I mentioned who with a spindle cell sarcoma with an NTRK rearrangement who was on, who's still on larotrectinib years later. So I think, and I really want to open it up for questions now, that there are a number of positive clinical trials. There are increasing treatment options for sarcoma patients, but it's really important to use all available treatment options. And
We lost the microphone.
does that work? No.
Yeah.
It does?
Yeah, that
Okay.
works.
So to obtain a better understanding of the biology...
No.
That's a sign for me to stop talking, right? But I think understanding the biology and the reasons for resistance to therapy, for response to therapy is really important, and that's where international collaboration is so crucial. And, finally, again, just thank you for all your support, and it's just a real honor to be here today. So, sorry, Marcus, that was a little bit longer than I thought it would be, and so I'm looking forward to your questions now.
We have a couple of questions over here.
That one was.
Up there at the top. Oh, here's one right here and then one on the top. Catherine's getting her exercise.
Exactly. I thank you, Robin, for an excellent presentation, a great overview. I was struck by many things that you said, but primarily by thinking out of the box next to what Gerry also said, I think. Could you perhaps elaborate on in which way patient organizations like our own could help you with that and how you collaborate with them?
Yeah, so that's a fantastic point. I think collaboration with patients is key, even to that sort of one-to-one interaction in clinic with a patient, in terms of saying, well, this isn't exactly the standard treatment, but the rationale for not using standard treatment is A, B, and C in terms of, well, we don't think doxorubicin will work that well. In terms of, disease that's confined to one limb or a lower limb, there's a good rationale for trying localized therapies such as isolated limb perfusion, injectable trials. So again, we're very fortunate to have, I'm very fortunate to work with Kevin Harrington, who has a number of immunotherapy trials and a number of viral vector trials where we actually inject tumors that are palpable, that are just under the skin. So the first thing is that sort of interaction one-to-one with the patient in clinic about why this approach, the rationale, so that the patient understands it. I think the second thing is in terms of patient groups and patient support, I think that's absolutely critical. I think, yesterday we talked a lot about collecting information. I spoke with Denise about, collecting information regarding people who've done really well in terms of off-label use of medication. So I think making us aware of people that have benefited from treatments that have been used off-label within a clinical trial is really, 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 really important. So, yeah, indeed. So it's all about communication, I suppose. But I think your role in terms of almost making a noise and making people aware is really, really, really important. Marcus, great.
We have
So Marcus and I have had good discussions in the past, so I'm looking forward to this. Oh, okay. Sorry. Yeah, I can. It's Marguerite, I think. My eyesight isn't that bad, but.
Can you hear me?
Oh, yes, great. I can hear you now, Marguerite.
Okay. Your presentation got me thinking about phase 1 results and emerging targets. How does your community of peers, you and other KOLs, how do you systematically look at phase 1 results across the whole gamut to identify where those idiosyncratic phase 1 responses in sarcomas that may provide a hint about future mechanisms of action that could be broadly applicable or applicable to certain subtypes?
Yeah, so that's a fantastic question, Marguerite. I think right now we don't do it in a systemic way, and that it's very ad hoc. I mentioned to a few people yesterday that we, about 16, 17 years ago, we analysed all the sarcoma patients treated on phase 1 trials at the Royal Marsden, and there were conventional chondrosarcoma patients that had durable benefit from drugs similar to azikibart in terms of not reaching that resist 30% shrinkage, but minus 28, minus 29%, and really, really durable benefit. But it takes so long to for that to sort of translate into a randomized trial. But in that case, I'm really pleased that it did. But I think that was more serendipity than anything else. And I think we really should pay more attention to patients treated on phase 1 trials. But I think it's really, really ad hoc right now, unfortunately. And it's challenging to try and think of a way to make that more standardized, just because of the fact that phase 1 trials open and close so quickly. At present, I used to be on the sort of phase 1 allocation meeting, and used to actually do the phase 1 clinic, but because of the fact that the sarcoma clinical workload is so high, I don't do that anymore. But I think we should revisit that. So I don't have a good answer to your question, just a rambling sort of answer that we should do it, but I don't know how. Hey, Marcus.
Hey.
Great.
No question. You should be not afraid. This is time.
No,
question.
I quite like it.
No, I want to make a comment to your question. Maybe a short example. We started some years ago in Germany a new process, building up a new clinical trial basis in Germany for all cancers. It's called the NCT, the National Center for Tumor Diseases. And this is a big project across Germany in different sites. And this will hopefully give us at the final stage 98 million per year to do clinical trials. IITs means early clinical trials, phase 1, phase 2, that are driven by the academic world and not by pharma. I think that's an important topic. But when we decided, together with our medical colleagues, this was a very interesting development because there had been 100 oncologists in the room, leading oncologists in Germany. And when we started to discuss about the priority disease areas, they had been not aware about the real problems of rare cancers. So we as patient advocates and our colleagues, we really advocated hard together. And I think that's the chance really to work together and really to advocate. And today now we have a priority disease area within this setting called rare cancers. And we also have a priority disease area called sarcoma. So we are now developing the first clinical trials within sarcomas. This means without patient involvement in a, I would say, professional way. It's not about noising and cheating and doing, but to work together with the colleagues and try to move the needle and really try to be strategic together to really change decision. This helps quite a lot.
Yeah, I mean, I use the word noisy just to raise awareness. And one of the things, well, just quickly, again, talking to Denise last night, you know, that analysis that we did of the sarcoma patients treated on the phase 1 unit with Johan and Stan K, you know, there's still one patient with metastatic Ewing sarcoma treated on a phase 1 trial of an IGF-1R inhibitor that is in complete remission 10 years after stopping the drug due to access. And again, it's such a tragedy that we don't understand why. You know, Denise has heard me go on about this. I've even got a slide on this, which it's crazy that we know that a subgroup of patients with that disease, with that very aggressive disease, benefit from this drug, but we just don't know why. Denise is yawning because I go on about that all the time.
Jeff, I think you have the next question.
So, I am
Jeff.
really impressed with what's going on in Europe with some microbiology advances and research that's going on. America, lately, not so much. And it could be political. It could be a lot of different things. But how do we bridge the divide and start working together and having our colleagues in the U.S. start working effectively with our colleagues in Europe?
Yeah, that's a great question, Jeff. Honestly, I could talk about this all day. I mean, even the... you don't want to know how much time Paul and I have spent on this in terms of the bureaucracy and the paperwork in terms of transferring radiological images, transferring tumor samples, in terms of tumor samples having one genomic analysis done in one institution and then being transferred to another country to have some different genomic analysis. It's really, really complicated, and in my view, unnecessarily complicated sometimes, because all of my patients just say, look, why aren't you using this tumor material to try and make things better for patients in the future? I'd say it is possible to do, but it just takes a long time to work through the the bureaucracy. One thing I would say, it's really important to have a team of people working with you. So, a dedicated person working on, the regulatory aspects of setting up a trial and a study. And I think, having a good team of people, it is possible to work through things, but the administrative hurdles, the bureaucratic hurdles are really really enormous. It's just, as I say, even within the UK, sometimes transferring information and material is really, really complicated, and as I say, unnecessarily complicated. But I think it is possible, and for the for the Tepentafus trial in clear cell sarcoma that Andrea Napolitano and Ivan Rosenbaum from Memorial set up, we're going to try and work on these sort of translational studies using the tumor material from North America and from Europe. But again, we have to work through the paperwork, and that's going to take a long, long time. Unfortunately, that's why my hair is completely white. Yes.
Robin, just a quick comment, and we'll go to Denise. I have to co-sign on that through PUSH.
Yes,
The amount of paperwork just to collaborate across globally is, the amount of legal fees we have paid just to try to move the needle is extraordinarily challenging. And it might be something as patient advocates that we can work on to change.
Again, to use the word noise, Marcus, I think it is really important, and your voice is so strong in that respect. I think the authorities, Sam knows, the authorities in the UK just view me as a troublemaker and a complainer. She's nodding. That's not the answer.
Denise.
So quickly, first I'll say I wasn't yawning. I certainly support and I feel terribly frustrated that we don't know more from the IGF-1 story 10 years ago. So many patients participated and people benefited, and we don't know why, and we just can't be in that place, and how we as advocate can change that. But my question that I have in mind that I want to ask you about is, you talk about the partnering, and I think about, as you say, there are even more characterizations or more subtypes of sarcoma, and partnering together advocacy groups with basic researchers and clinical researchers. Are there enough out there? So I think of the clear cell sarcoma and working with and share this, that to try to find champions to work with you to focus, because there are so many different subtypes, and finding clinicians and basic researchers who will focus their attention on these super rare subtypes, because we're not going to make progress alone as advocates, and we need to have that partnership, and it's just a struggle. And I don't know if you have some advice of how we can better address that issue.
Well, one of the things is actually being at this meeting and seeing all of you. Honestly, the sort of direct communication is so good and really, really important. And I think, again, Denise, I think it's collaboration, and that we need to all work together, because it's. Again, the heterogeneity is just so profound, but I think we just have to work together. And, it's probably, there's not a one solution for all of these different diseases. We'll probably have to just use different approaches for each individual disease. And that's the really sort of challenging thing, isn't it?
So many hours in the day and so many specialists, and you're being pulled in so many directions. And we appreciate that and understand, and yet the frustration on this side of trying to, recruit somebody to be an advocate with you, and it's a challenge.
Lenny.
Hi, thank you. Okay, I have 10 things in my head between Marcus, Denise, Jeff, what everybody said. First, I want to say thank you for that paper you put up there about the systemic treatment for clear cell. You have saved more lives with that paper than you even know. So thank you for that. we don't see that very often anymore, given especially to the children, and patients are living longer and living because of it. So for everybody here, just so you know, reporting even negative data or something that's not a positive, okay, this is a good treatment, but by saying this is not effective, you can save lives. And Jeff, as far as this goes, I do think we can make change, like Marcus said. I think that we have that ability, and we're working on that. We've got an AYA group in Clear Cell we're putting together that we plan to take to speak to people in DC. We can talk more about that. I also want to thank you for what you said at CTOS years ago in the debate about everybody should probably go into a phase 1 trial, and you had up here on the screen. Denise remembers, as a mother, I hate that, but as an advocate, it's the only way we're going to make better. We've got to do this. And thank you for helping us get the phase 1 trial. You did more for getting that done in the US than we were able to do in the US, and I appreciate it. But my question, and I guess what I'm saying, is what we struggle with, I think you know this, is that phase 1 trial. You were on the call when we were developing it. We had the FDA. I didn't know anything at that point. I still don't know much, but I know a lot more. We couldn't get the endpoints that we needed.
I distinctly remember that call, and I distinctly remember the FDA were not too happy. Sam, they were even more unhappy with me than nice, but anyway.
But you know what? It got it done. We got it done. We passed the phase 1 safety study. The problem is, they speak out of both sides of their mouth. We can't have that endpoint because...
You can say that.
Well, I'm saying it, and Jeff can say it.
Let's make sure that's not on the recording for Robin's.
Well, I said it, not you. But we have to say that, and we have to speak a loud voice, because I don't know, Denise can speak to this better, but if we don't keep pushing, it's not going to change. And we have to keep pushing in a way. We have to keep pushing back, because there's no success. We are having no success doing it the way it's been done for so many decades. They told us on that call that we could not change the endpoints, period, and that we were only doing a phase 1 trial. They were not going to look at efficacy. The NCI refused to take on the phase 2, even though we passed all safety studies, because they saw no signal of efficacy. You can't I'm calling them on this. You cannot do that. We're going to change these endpoints. We're going to change the way phase 1 trials are run for ultra rare. So.
And it's great. I totally agree. And your voice is so important in that. Your voice, honestly, is so important in that. The other thing, just before I forget, and it's a way, we discussed this a little bit yesterday. I'm going to pull Denise's leg again and tease her a little bit, but the sort of academic structure, the way that things work, we have to almost play the game in terms of grants and publications. But I think there is a way of doing that, and that's why I mentioned the Accelerator Consortium, which is more of a long-term thing than a clinical trial of treatment X versus treatment Y. That will be a huge resource for not only for us, for Paul, but for people in the future, in terms of collecting all of the radiological and molecular and even the sort of pathological data, in terms of what is and Judith is still here, I think, what does pathological response actually mean in different types of soft tissue sarcomas. So, having both sort of clinical trials, but also long-term projects. And again, Gerard and Roger have been so helpful in terms of, giving advice regarding that grant. And, in terms of getting more peer-reviewed grant funding from the original CR UK grant, so that the project is actually sustainable, because for that sort of thing you need years in terms of not only working through the bureaucracy, but also in terms of actually analysing all of this information, which isn't a sort of rapid readout. So just to say thank you to both of you. I think Roger was expecting a few questions from you, Roger, but
Roger's been quiet.
No, I'm teasing him a little bit. He's used to me, so. No, you don't. I talk too much. I had too many slides, so.
We have a question over here, and then maybe this is the last question.
Oh, pressure.
I-
Okay,
Make
I'll
it
try to make
good,
it
make
good.
it
All right, well, thank you. Oh, over here.
Oh gosh, sorry.
No, that's all right. Thank you. I
do need
really,
glasses.
yeah. No, I appreciate the creative approach and really trying to look at different things and off-label for patients. And I wanted to get your thoughts on how we can maybe change the perception even of I think oftentimes patients enter clinical trials very late in their treatment journey, which kind of, in a way, sets up these trials for failure because these patients are coming with often unresectable disease, heavily pretreated, and we know in immunotherapy, I think there's evidence that shows that if you're either also harvesting their T cells to make the cellular product late, the quality is lower, and certainly the immune response is going to be not as great. But probably even for other things besides immunotherapy. So thoughts on changing that perception of having patients enrolling, like you said, that everyone should be doing a phase 1 trial earlier in the process. I think there's some examples like there's the MAP plus Cabo trial for osteosarcoma in the US where it's, up front for the, frontline therapy. But thoughts on that and perhaps maybe even how risk stratification can help with that? Because I know there's probably clinicians don't necessarily want to put their patients on experimental drug early on when they know that standard of care will work for a large percentage, but I think it's really important to maybe get those trials earlier.
Yeah, again, that's a fantastic point and fantastic questions. Again, I suppose it's sort of context dependent in terms of presenting somebody with the option of a phase 1 trial or early involvement in a clinical trial versus standard treatment. I think, as we've discussed, for sarcomas such as clear cell sarcoma, given that the standard treatments that we have available are not very effective, I would always present it to somebody and say, well, this is the information we have. This is information we have from retrospective studies. We can always fall back on standard treatment later on, but it probably makes more sense to go for a clinical trial early on. I think the chondrosarcoma trial of the death receptor 5 agonist. There's a real benefit for patients treated in first line compared to later lines because, as you point out, it's not that the previous treatments have been effective, far from it. It's because the cancer has just grown so much and that it's so complex and bad by the time somebody enters a clinical trial, 4th, 5th line, that it's unlikely that any treatment will work. I think there are different views on this. Some of my colleagues, and I'm not sure if Peter's in the audience, but some of my colleagues would say no, you always have to treat with doxorubicin first line, but I would argue with that for some of these histologies it just doesn't make sense where we know that those drugs just don't work. Forgive me, your second point was how can we? Yeah, so. Yeah, I think right now it's still very very crude in a way in terms of histological subtype and a sort of a one-to-one discussion with that individual person. So I think a lot of work still needs to be done on that. One of the points that I always try and make is that the preoperative setting is also a good scenario to assess a new drug, particularly if there's a sort of window where the surgery is going to be not in the immediate future, but is planned for 2-3 months' time or something. Then assessing a drug in that setting is really useful because you can capture the radiological information, the clinical information, but also information regarding what happens to the tumor and the effects of the treatment on the tumour and the surgical specimens. So I think you're right, that would be a much more rational setting to explore and evaluate new drugs rather than fifth, sixth, seventh line treatment, which you know, for a lot of cancers by that stage, it's so complex.
I know I said that was the last question.
I
But
should have
since we
definitely
have
cut
Robin
those
here,
slides.
not often don't have this opportunity, we are going to have one more question back here in the back.
Hi there, Andy French from Sarcoma UK. Hi, Robin.
Hi,
Excellent
Andy.
talk in the session. It was just really on the drug appraisal process. As you mentioned, in the UK, it's quite difficult at the moment. And having been involved with colleagues and trying to bring in the kind of clinical evidence, working with healthcare professionals and patients bringing in their lived experience, there seems to be three issues. One's on the cost, which is kind of quite clear. And recently, when ripretinib was approved, the government just basically raised the threshold. So that seems to be kind of important. The lived experience seems to be, my impression is, a bit of an afterthought, really, in terms of the weighting of in their decision making. The other thing is on the data. So the low numbers and the particular model that NICE are taking. And I just wondered if there's anything, and this isn't a criticism, but in terms of the international work you're doing with colleagues and the kind of study trials you're putting together, is there anything that you're thinking ahead in terms of how you construct those trials to kind of, I suppose, to kind of play the game to try and get things through so that when we get a great treatment, it stands the best chance? And, you know, I'm saying that from the point of view that I think NICE have a bit a flawed model at the moment.
Yeah, that's a fantastic point, Andy. And Sam's looking at me, so maybe I'll ask Sam to help me answer this question. So Sam's point is in terms of collecting quality of life data and trying to compare you know, different trials, desmoid tumor with soft tissue sarcomas, which is I'm not sure if I'm allowed to say that, but that's a sort of discussion that we had with NICE. So I think it's really hard, and I think, you know, this almost, this slide speaks for itself in terms of a drug being effective, and the patient advocates in the larotrectinib appraisal, you know, they've all done so well on the drug in terms of side effects that I suppose there's a balance there, but you know, NICE is almost asking for another clinical trial in a way to. And tell me if I'm wrong, Sam, in terms of I'm being. But. Yeah, but I'd argue that the ripretinib trial had quality of life analysis included, and you know, there's been a separate publication on benefit in terms of quality of life. So, you know, there's only so much information in a way that you can collect from a clinical trial, and I think, as I say, just from my experience with larotrectinib, that patient that I mentioned who's been on the drug for about 5 years with no toxicity, you know, I almost think it's unethical that she had to go through standard treatment, again, knowing that it doesn't work, but that was the only way we could get access to larotrectinib. So NICE don't like me a lot, as Sam knows.
Sounds like a lot of people don't, Robin, from what you keep saying.
Yeah, that's true.
We do.
I think Sandra had a question as well, so.
It was probably more of a comment, if you don't mind me saying, just in response to what Christina was asking, is about how do you get new drugs into patients earlier in their journey. I would say involving patients in designing those trials really makes a huge difference. In the Ewing's trial, we are introducing a new drug for patients with metastatic disease into first line, and that trial was designed with patients to kind of join us. And so I think you can do it, but it has to be context dependent, and what is and knowing that these players, that the outcomes for patients are poor, and you do know that at the beginning. The stratification is not good enough yet. We do use such crude measures. Hopefully kind of circulating tumor DNA and other things like that will be able to pick up patient groups better than that we can add more questions up front. But then, of course, even if you know someone's got a poor outcome, it's like, what are you going to do about it? And so the next thing. I just wanted to say one other comment about the importance of the negative data or understanding the landscape. So I echo what Robin says about clear cell sarcoma. I think it's the wrong thing to do is to give the patient doxorubicin, and I would say that quite up front. We are just finally in 2026 getting access to pazopanib for our patients because it's become generic and the cost has gone down 90%. But the license indication is for patients who've received one line of chemotherapy. And so we had to argue with our trust, this is on a completely local level, that patients should not have to have chemotherapy to get the drug. But we could find papers and even guidelines saying that these are chemo-insensitive. But it's that data that you need to support those arguments. So we're very grateful going through. It's like, here's another paper. But we need that information, the negative information, just as much as the positive information.
I think that's absolutely right. Oh, sorry. Sorry, I'll finish.
So one comment I do want to make before we wrap up is the magic of SPAGN is that we all come from very different areas. Some of us very privileged, some of us not. And we've had this whole conversation about access to clinical trials. And from the US, I'm from one of the most privileged countries when it comes to that. But we have friends in the room that I had the joy of conversating with since we've been here that absolutely don't have this. And so we have to remember it's great that we can all come together and have these conversations, but as we're doing our work as well and talking about access to clinical trials, and we're focused on what's happening in our own countries, we also, and I mentioned this yesterday when we were talking, have to carry the water for our friends and have to help each other and understand that we're on a wide spectrum, and it's wonderful to be able to come together here at this conference and come together and have these conversations and learn about, but also remember not everyone in this room has the same access that some of us do. And that is, as patient advocates, we work on so many things, but that is one thing we have to work on together, and this is a magical place to be able to do it, but it doesn't leave here. Not that the whole show is ending now, but I just wanted to make that comment. But again, thank you, appreciate your patience as we went over, but we did start late because of Denise, so there's that. Anyway, now we really do have a coffee break, so thank you so much.